Good morning. Myself, Payal Sector Research Scholar, Department of Agriculture and Food Engineering, IIT Kharagpur. Today, I will be dealing with a small topic. My lecture session is on anti-nutritional factors. So, when we usually the anti-nutritional factors are mainly present in the plant sources. So this is one topic which usually we have commonly it is some questions, one or two we will be getting in GATE. Sometimes it comes in the other competitive exams too. So what is the idea here? The idea over here is I'll be just giving a brief outline about the anti-nutritional as a like a sum up. Okay, anti-nutritional factors. So this lecture will deal with the different types of anti-nutritional factors. Why it is important to know the anti-nutritional factors? Because sometimes we work in extraction. Sometimes the research goes with extraction from a plant material, or you are about to go for some product development from a source. May say suppose even you have example, some soya based product you want to make, soy based. So they have certain anti-nutritional factors. These anti-nutritional factors you need to identify and you need to deactivate or remove it, inactivate, or you have to remove that. Then you can come with you can go further for your work. Okay. So just a brief idea, what do you mean by the anti-nutritional factors and what are certain anti-nutritional factors, the common ones which we can discuss here. There is a wide range of anti-nutritional factors. In that, the important factors which important anti-nutritional factors are trypsin inhibitor is a very important one. Usually, even in I'll tell you, ASRB net food technology, that is code number 55, a question came. Soybean is having the anti-nutritional factor present in soybean. This is a question in ASRB 19 net 55 is the code, that is food technology 2021. The question is ASRB net 2021. The anti-nutritional factor present in soybean is, okay, and the options were there: trypsin inhibitor, it was saponin, it was phytate, and one more option was there, probably tannin or goitrogen. So the answer will obviously come as trypsin inhibitor. So this is one of the questions which was there in the competitive exam. So you can just, I'm just underlining this, you can take this point. Now the second point is about the after trypsin inhibitor, we are having the phytates, we have the tannins. We have phytates, we have tannins, we have lectins, okay, oxalates. So these are some of the anti-nutritional points where we can, anti-nutritional factors which we can, even goitrogens will be there. Even you have saponins. Okay, so these will be your some of the anti-nutritional factors. Okay, now what do this anti-nutritional factor, what does they do? They just get in the interfere with the other nutrients that is present, such as they will interfere. Yeah, so they will interfere in between the nutrients, okay, like the proteins, the minerals, calcium, zinc, iron, iodine. They will interfere with that and they will not allow them to function properly. This is the difficulty what anti-nutritional. So what they do is the anti-nutritional factors will go and interfere with the, they will interfere with the nutrients, interfere with the nutrients, okay, such as zinc, calcium, iron, iodine, and so on. Okay. So the next one is they interfere with the nutrients. Is it first thing? They will interfere. Then what will happen if they interfere with the nutrients? They also reduces the digestion and utilization. Reduces digestion by some. They will interfere with the nutrients. Thereby, what happens? The utilization of the nutrients is not proper, as well as it reduces digestion. So, what are the symptoms we are getting? We are getting nausea, we are getting headache, we are getting nausea, headache. We will get some rashes. So these things keeps occurring. So that's why it is important we should remove this anti-nutrients, and then we should work on the any such research, or you are working for producing a like formulating a product, but these anti-nutrients are necessary to be removed, especially if it is a plant-based or plant-oriented product. Okay. Now, trypsin inhibitor, as I told you, there was a question in ASRB net 455, that is food technology in ASRB net 2021. There was a question about the anti-nutrient present in legume, and I said you about the trypsin inhibitor. Okay. So one such big example about trypsin inhibitor present is soya bean. Besides soya bean, we also have some other like lima bean, kidney bean. Okay. We have egg white. These also contains the trypsin inhibitors. So you should be aware of at least, you can say the legumes mainly, the proteins. Mainly the legumes will have the trypsin inhibitor. Along with that, exceptionally we can say egg white. Okay. What do they do? The trypsin inhibitor, they will compete with the protein. So whatever proteins you are, obviously all these legume-based egg white, they are rich in protein. So the proteins, what is the rich over here egg white? Albumin. You are aware of it. So when I'm giving some key points, please do take that. It will be helpful. So we are talking about anti-nutrients, but again, some protein we are like what is soybean is having some some protein. Lima is having some protein content. Kidney will be having kidney bean actually. Okay, and egg white is having albumin. So in general, it has been said these proteins of this particular sources, they will be, the trypsin will compete with the protein. Okay, competes with the proteins to bind to trypsin. They will bind with the trypsin. Okay, so they will combine and go with the trypsin part. That is how it is happening. So inhibit the activity of trypsin in the gut. So it obviously there will be a problem in like interfere with the digestibility of the dietary protein, and they reduce the utilization. So in the previous just a minute before, I have said that they are interfering with the nutrients. They are reducing the digestibility. As a result, it is causing some symptoms in the body like nausea, headache, and so on. Similarly, if I'm talking about the trypsin inhibitor, the sources of trypsin inhibitor are the soya bean, the lima bean, the kidney bean, and the egg white. These are the sources. And then what we are talking is the protein will be, they will compete with the proteins which are present there, and they will bind with the trypsin. The trypsin inhibitor will compete with the proteins. They will bind with the trypsin. As a result, the trypsin will be getting inactivated in the gut. So it inactivates, obviously it will inactivate because they are just getting binded with they will inhibit the functioning, the normal functioning, and interfere with the digestibility of proteins, dietary proteins, and reduce their utilization. So this is about the mechanism or how the antinutritional factor, that is trypsin inhibitor, is affected. Now the next one is like heat labile. The extent that heat labile, if you are having, if you are autoclaving, okay, if you autoclave or if they are usually heat labile, so the extent and ease of heat inactivation varies. So they are heat labile, but if you autoclave, they will improve the utilization to a certain extent. If you autoclave the particular material, mainly if you say suppose you are autoclaving the soya bean, it will inactivate the trypsin inhibitor to much a greater extent. Boiling soybeans, if you say, if you are boiling it for 14 minutes, or you can say around 14 to 20 minutes in range, definitely it inactivates 80% of the inhibitor. Okay. If you do for 30, so time temperature combination, if you do it, if you try to boil it for a more number of time, like number of hours or number of minutes, the time duration is higher, then definitely it will reduce. The percentage will be more. The percentage of trypsin reduction, trypsin inhibitor inactivation will be more. Okay, so this is how we go with the trypsin part. So autoclave and boiling is an option from where you can inactivate the trypsin. Okay, I mean inactivate the trypsin inhibitor rather, the anti-nutritional factor. The next part we will be talking about is the phytate. What is this phytate doing here? Okay. The next anti-nutrient we will talk about is phytate. So what does phytate do? Phytate will be mainly present in the seeds, legumes, grains. Seeds, grains, nut, legumes. Okay, so it is there. Now, unrefined cereals and millets. If the cereals are not refined, unrefined cereals and millets, so they are a good source of phytate content. Good source of phytate content means their anti-nutritional factors are high. Okay, so they store phosphorus as phytic acid and in their acid. Okay, so the phosphorus will be, they will be taking this phosphorus part, which is the phytic acid. Phosphorus as phytic acid in the husk in the form of phytate salt. Phytate salt. Okay. So this phytate will be, you have heard about the term inositol, right? So the phytate is hexophosphate of inositol that acts as a source of bound phosphorus, the seeds during germination. Okay, so the hexaphosphate, like phytate is hexaphosphate of, phytate is hexaphosphate of inositol. Okay, so this particular part we can say. Now what does phytate do? They have a bind. Now in the first, as we have introduced the class, I said they interfere with the nutrients, available nutrients. So here the phytates have a strong affinity. The phytates have a strong affinity to bind, or what they do is with iron, zinc, calcium, magnesium. These, they will be having strong binding affinity towards this, right? So what happens if they are binding this? They will be, when the particular phytate is getting binded here. So what happens? They result in insoluble salts. So resulting in insoluble salts, which will land up to what? They will be giving you, they cannot be, you know what, you cannot, bioavailability will be lowered, decreasing the bioavailability of these nutrients. So what the anti-nutritional factors are doing? They are just reducing the bioavailability of the nutrients. They are binding with the nutrients, reducing the bioavailability, as well as they are interfering with the common nutrients which are required for digestion and all. As a result, it creates trouble. Yes. So if you do germination, then the phytate content has a considerable amount of phytate can be reduced during germination. This is your second part. Then we will talk about tanning. Okay. If I'm talking about tannin, this will be my third. Already I have spoken about tannin is the third one. Okay. So the third one is the tannin, major group of antioxidant polyphenolic compounds. Where do you find tannins? Mainly with tea. You get tannins, right? So here what is happening? Uh, the tannins are mainly we will be getting in tea, that is green tea, and even if you be more specific, it is green tea. We get it in tea, we get in tamarind, spices, legumes, ovio, legumes, etc. Okay, even some fruits and vegetables is having. What they do, they specifically bind with the iron. They will bind with iron, okay, and interfere in the iron absorption. They also bind with proteins, but mainly it is iron absorption. Is it okay? And reduce their availability. So this is about the tannin, the sources and what they do. They mainly go and make it. So why this is like given in this manner, you can understand that what are the, if we are asking, what are the some questions you are getting. Anti-nutritional factor present in the tea. An anti-nutritional factor that binds with iron or it reduces the iron absorption. So the immediate answer you will be going with is your tannin. Okay, so this is the idea of taking in a very crisp manner what all can be the probable points you should remember. If you follow this video, you will be understanding follow this lecture, you will get just the crisp points. Okay, the crisp points will be helpful in your some competitive exams as well as for your some interviews and so on, mainly for your exam purposes. That is for when you go along with it. The fourth one which we are talking about is lectin. So we have discussed about the tannin, we have discussed about the phytate, we have discussed about the trypsin inhibitor. The fourth one is about lectin. Lectin is mainly the source we are getting it in tomatoes. We will be getting it in peppers, eggplants, even legumes. So the source we can say lectins in tomatoes. We have it in potatoes. Okay. This is number one. Number two, we are having it in even the legumes. This lectin, if it is present, it causes flatulence. Okay. They inactivate. If you want to inactivate it, you can use heat, fermentation. Suppose you are fermenting a particular legume. So to an extent, the lectin gets inactivated. That is the meaning here. Okay. Then oxalates. Oxalates and goitrogens we will be discussing, and the general, so fifth one is oxalates. Oxalates are soluble, that is forms soluble, that is potassium and sodium. Okay, or insoluble. See, oxalates are in two forms. One is the soluble form, that is, I will omit this and I'll come to the new fresh page. Oxalates. So what is oxalates? The two things we have. One is the soluble, other one is insoluble. So insoluble salts, if you take, okay, or esters, they are the salts, soluble salts or esters, and insoluble salts or esters. If the soluble salts or esters is sodium and potassium, if you talk about insoluble salts esters, you will be having calcium, magnesium, iron. Okay. Now, they are mainly the oxalates you will be getting in the green leafy vegetables. Green leafy vegetables and some legumes you will get. They, what do the oxalates do? They interfere with the calcium absorption. They interfere with calcium absorption and by forming insoluble salts. So they will interfere. The oxalates will interfere with the calcium absorption. Okay. And insoluble salts cannot be processed out of the urinary tract. Right? You are aware of it. So when they are interfering with the calcium absorption by forming the insoluble salts, and this definitely cannot be processed out. The insoluble salts cannot be processed out from the urinary tract. This is the problem that happens. Okay. So they will be interfering mainly with the calcium absorption. Right? The other one which I told you now, that is mainly with the another explanation I told you about tannins, that is going with iron, mainly the iron binding. Here it is calcium binding. Okay. Next is about goitrogens. This is the sixth one. The goitrogens are basically the thyroid hormones. By interfering the iodine, they will mainly go for the iodine interfere, or they will bind with the iodine. Okay, mainly with the leaves, vegetables, leafy vegetables and leaves, you can say. That is cauliflower, radish, all that. Okay. Next is anti-nutrients are found in almost all foods. So here I have mainly figured out the main six that goes with the trypsin inhibitor, which is binding the protein, competing the protein and binding the trypsin actually. Then we have discussed about the phytate. The phytate is mainly dealing with the bioavailability, mainly with it, mainly with iron, and then obviously with zinc and calcium, magnesium also gets affected. Okay, then the tannins are mainly binding up with the proteins, that as well as iron is the main nutrient which it gets binded with. Lectin is mainly, it is binding, like what happens in lectin, it is causing flatulence. Okay, and if you take it taken in the raw form, and the oxalates are mainly for calcium absorption, they inhibit and interfere that. And when you talk about the goitrogens are for the iodine, mainly the iodine is getting interfered here. So as a result, you will be having some goiter issue. Thyroid stimulating hormone, the trigger the pituitary that will release the thyroid stimulating hormone, giving rise to your goiter. This is the idea. Now the anti-nutrients are found at almost levels in food. So the suggested or the methods which we can say, the traditional method, mainly we can soak it to remove the phenols, total phenol, the tannins, we can do, and phytates. We can remove this in that way. One is the soaking method. Germination also we can say, germination, fermentation, methods to reduce anti-nutrients. So soaking, this is one thing. One, two, germination. Three, fermentation. Okay. Then another one is heating. We can say heating or autoclave, or you can say sterilization. So these are the probable traditional methods which can be suggested to remove the anti-nutritional factors or anti-nutrients from the food samples. So with this, I complete my session on the anti-nutrients. The, as I mentioned, the idea of taking this small topic is what are the in order to get a small insight. So the outcome of this lecture is small insight on what are the types of major anti-nutrients present in the food, how do you reduce that, and which anti-nutrients mainly affect what type of nutrients. Like, say for example, if you say about if you say about trypsin inhibitor, definitely the trypsin inhibitor is competing and it is binding the trypsin. So like that, if you talk about oxalates, they are mainly affecting or they are interfering with the calcium. So these type of important factors you can take as a keynote, takeaway home points or takeaway points, you can take it. So this can be highlighted because certain cases, one or two questions you may get like MCQ or so from this type of topic. Is it clear? Thank you for listening.